Okay, can you see my screen?
Yes.
Okay, my name is Chaochun WEI from Shanghai Jiao University, from the Department of Biomedics and Biostatistics, School of Life Science and Biotechnology. So first, I would like to thank Dr. Yu for giving this opportunity to talk about our work in this topic. This year is the 20 years anniversary for the initial human genome project, the initial human genome. After 20 years, I still talking about this incomplete human reference genome, and now it's incomplete annotation. It's a little bit insulting to those people working on this region, but I think it's okay. I am also working on this area. First, I will give a little bit of background, then I will talk about the human pangenome. That's also reflects the incomplete of the human reference genome. Then I will talk about the human genome annotation, my own opinion about the human genome annotation. This is the figure that from the paper last month about the legacy of the human genome project, starting from 25 years ago. Then first initial release of the human genome project as human genome sequence in 2001. Then for this year, I think last month, it's called the first full genome sequence completed. That's from end to end human genome available. But if you look at the paper, it's about one individual. It's not for everyone. So even with this one individual genome available, there's still a lot of space to see the completeness of the human reference genome. This is another paper talking about the 20 years anniversary of the human genome, but it shows the human reference genome still have about 150 million bases missing in the reference genome, and especially those centromeres and telomeres, and some of the region that's folded very closely, then they cannot be easily sequenced. So in the human reference genome, about 150 bases missing. Even with this, the fully sequenced the whole genome that's fully sequenced that individual, they filled a lot of gap, but still for some people like me, maybe it's not from the same population or ethnic group as the full genome sequenced. So there are a lot of so-called underrepresented populations. For example, the human reference genome itself, if you look at the ancestry makeup, majority of the 20 or 21, 22 individuals that contributed their samples to the human genome project are with the European ancestry, and 37 African ancestry. With East Asian ancestry, only 6% in the human reference genome project. And also in the available data that statistically in the GWAS analysis, the population in different population are very unbalanced. So even with the full length, so-called end-to-end human genome available, there's still a lot of genetic information out there. So I will talk about the Han Chinese pangenome that's we have working on for a few years. The pangenome is the whole genetic information of a population. So it's first introduced in bacterial genome. Most of, or almost all of the biomedical research now based on genome and based on the reference genome as the standard reference to do the analysis. The pan-genome concept, on the other side, is focused on the divide of the genomes into core genome, dispensable genome, and a unique genome. For example, the genome that's shared by every individual in that population is called core genome, and shared by some, but not all individuals are called dispensable genome or valuable genome. And only one individual have this region, then it's called unique genome. So this concept was first introduced for this called group B streptococcus. And after they combine 10, 12, or 17 different strains of this bacteria, then they found this bacteria has infinite number of genes. That time was very surprising. So intrigued me to ask her, how about in the human genome? If we have many human genomes considered together, what will happen? Well, we have infinite number of genes, something like that. But that time, 10 years ago, the 1,000 human genome per year is already available, but the data, the human genome, the data was low coverage sequence data, and the assembled genome might be not that good to make a reasonable conclusion. So we shifted to the rice genome. In that time, we have 3,000 rice genome available that time. About 400 of them are high quality, high sequencing depths. So we use that data, construct a pan-genome construction and analysis pipeline, and use this pipeline to analyze the rice genome using 3,000 rice lines. And we found a very surprising conclusion that rice as a species, its pan-genome is about 70% bigger than an individual genome, which is the reference genome. The reference genome was about 380 megabase pair bigger. The size of 380 megabase pairs, and the pan-genome will have 268 more new sequences than the reference genome, and 20% of important genes, which we used to believe that they should be core because they are important. But we found 20% of those important genes are dispensable. Actually, they show in some of the individuals, but not in all of the individuals. So it comes back to the human pan-genome. What's the situation for the human if we have a human pan-genome? So we spent 2 years to expand the pipeline that work for the rice genome to construct human pan-genome. Because the human genome is about 10 times bigger than the rice genome. So we have this called HuPan for human pan-genome construction. Then we constructed a Han Chinese pan-genome that's derived from 275 Han Chinese individuals. Then we found in total 33.5 novel sequence that's both sides cannot be aligned to the human reference genome. There are another about 48 something that's partly can be matched to the reference genome, but it's still novel. So I will focus on this. Both sides cannot be aligned to the reference genome. So 33.5 million base novel sequence. Among this, about 4 megabase actually can be matched to the patch sequence of the reference genome. For the remaining 25.9 29.5 megabase sequence, about half of them actually can be matched to the African pangenome or the under the SGTP. So we think those regions, although we found in the Han Chinese pangenome, but it's actually shared by many other ethnic groups, but they were not in the reference genome. So in total, we found about 165 novel genes, and we believe that 40% of them are Han Chinese specific. So this is we later found that we overestimated, maybe overestimated novel genes because a lot of them are repetitive region enriched genes. So after we remove those repetitive genes, there may be much less smaller number of the novel genes, but still there are some novel genes in there. So we in last year there was a Salzburger from Johns Hopkins that he have published he published a paper in Nature Review Genetics, summarized the size of the human pangenome. So our work was showing here, saying about 1%, which is 29.5 megabase, is not in the reference genome. And he actually worked on the African pangenome, which is derived from 910 individuals with African ancestry. So his group has 296, close to 300 megabase, which is about 10% of the size of the human reference genome. So in summarize, I would say the novel regions will be about 1% to 10% of the reference genome are relatively new to the reference genome, which is not in the human reference. So the question is, this 1% to 10% missing sequence in the reference genome, are they important or not? So we searched some literature and found the Iceland genome sequencing project. They sequenced about 300,000, the whole population of Iceland, and there was a segment of 366 base pair insertion that not in the reference genome. It's significantly associated with myocardial infarction, with heart attack. So which indicated that the region that's missing in the reference genome might have important functions. So this is the summarize of my first part of the talk. The human reference genome is still incomplete. About 1% to 10% are missing compared to the human pangenomes. Missing sequences may have functions. So take-home message for this part is we need to use pangenome instead of the reference genome for the as the standard annotation and standard reference sequence for your biomedical research. So the second part is I will talk about annotation of the human genome. I don't think I have many time left, so I will go quick. Gene structure, patterns of genes. Oh, this seminar is about the AI in so we did use machine learning technologies for the genome annotation, the gene structure prediction. So there are a lot of patterns, machine learning technologies involved. So this is a typical human gene structure. If you look at that, the intron region is 10 times larger than the exon regions inside a gene. If you look at the genome for genes and intergenic genes, you will find that intergenic gene region will be 10 times bigger than gene regions. So the coding region of the the human genome is very difficult to find actually. But the question that how many genes do we have in the human genome, until I think until now, it's still an open question. In 2018, there's a paper also published by Salzburg that he asked how many genes do we have. So if you look at the databases available now, like ENCODE and the GeneCode Ensemble, RefSeq, and CHESS, the protein coding genes number are close to 20,000. That's pretty consistent in different databases. The number of non-coding protein genes or other genes in total, in terms of transcripts, however, the total number of transcripts is about the 200,000, or the CHESS have 300,000. So, but not all of them are protein coding transcripts. So if you look at the protein coding transcripts, that traditional meaning of gene, that's less than 100,000. So I will skip this history of gene prediction in the human genome and my work on the human genome annotation. Go back to the number of protein coding estimation first directory. So we did develop called the alternative splicing prediction system called OutScan, and predicted on the human genome, which is we extended it from the generalized hidden mark model, and they use a technology called multi-layer beta B algorithm that's developed by my group. And we, compared to the other method, looks like it's pretty good compared to the other method. And we validated with the prediction. We actually predicted 360,000 with non-redundant transcripts. But with the iron-seq data filtering, go back to 7 years ago, we have 31,000 that can be validated by iron-seq data, but the splicing side is not included in the database that time yet. And we designed PCR validation, then tested on 20 tissue, 20 cell line, and 8 RNA samples. Overall, the success rate is 90%. So we concluded that about 30,000 novel transcripts can be predicted by my group in our result. And we also compared to the mass spectrum data, and looks pretty good. We have like 36 normal protein validated. So overall, let's come to the end of my presentation. So overall, we estimated the number of protein coding transcripts in the human genome is larger than 200,000. So this is a take-home message for you. More than 50% of human proteins are not annotated yet. That's the end of my presentation. So we estimated more than 200,000 protein coding genes in the proteins or protein coding transcripts, 200,000. And in the database, if you look at the gene code, there's only about 78,000 now for full-length protein coding. So I will end here. This is my group, which did most of the works I mentioned here. So I will stop here and take questions for 1 minute or 2 minutes.
Thank you, Professor Wei, for the presentation. Sorry for not introducing you before. I had some issue with the sound. I couldn't hear anyone. So I will do that now. So Professor Chaochun WEI is a professor of bioinformatics and biostatistics at the Shanghai Jiao University. So we have a couple of minutes for quick questions, and we will have a question session at the end. So please, if someone has any question. Maybe I have a quick one before we move on. For many researchers, using a reference genome as almost a daily tool to do alignment I still see a gap in how to use it practically. So there are a few, we are still using a classic reference genome and classic aligners to do that. So what do you think is missing to really use pound genomes as a daily tool for aligning our new
that's an excellent question. So theoretically, pangenome now can incorporate every individual genome into the pangenome, theoretically. But if you want to use it as we use the human reference genome, there are still a lot of tools, including visualization tools, alignment tools, and like gene prediction, annotation, comparison. There's a lot of tools need to be developed. So it's not an easy task. We have working on that for 3 years, but it's difficult. As you said, especially for biomedical tools for different professions like doctors, they even for traditional methods, there are still a lot of difficult. Yeah, but for the pangenome, it's still maybe there are still a few years to come.
Okay, thank you. Okay, so for the sake of time, I propose that we move with to the next presentation, and please think about questions for the session of question and answer at the end if you have some. Thanks again, Professor Wei, for your presentation. So now we will have the next talk is Professor David WEDGE, who is a professor of cancer genomics and data science from University of Manchester. Thanks, David, for being here. David will be talking about the genomic evolution of prostate cancer. So that's one of that's the first talk today about cancer evolution. I think the next one will be also about this topic. So please, if you can share your screen and start your presentation.
Thank you. I hope you can see my my screen. First, I'd like to thank the organisers for the invitation to speak at this meeting. So I'm going to be talking about genomic evolution of prostate cancer. It's often stated that cancer, like almost all biological processes, is an evolutionary process. So cancer growth is driven by driving mutations and is subject to environmental effects such as hypoxia in the surrounding healthy cells, drug treatment, radiotherapy, and environmental exposures such as the well-known ones such as tobacco smoke and UV light, but also there are kind of rarer exposures which drive the evolution of cancer. So cancer evolution means kind of different things to different people, and so we wanted to look at evolution from a number of different viewpoints, including what you can see on this slide. So inter and intra-tumor heterogeneity, response to treatment, including drugs and radiotherapy, drivers of metastasis, and maybe most importantly, the trajectories or pathways to progression in cancer. The data that we used in this study came from the International Cancer Genome Consortium, or ICGC. You can see on this slide that the ICGC is actually a very large international consortium composed of a large number of different projects from different countries. It's quite noticeable. You'll see from this slide that after the United States, the 3 countries that have contributed most projects to the ICGC are the 3 countries actually represented in this meeting: China, United Kingdom, and France. But the data that I used in this project was from one specific project, which you can see with the red oval, was the prostate cancer study from the UK. I should say I'm not going to talk about this today, but we have since publishing this paper, we've actually joined together with prostate cancer projects in other countries to form an international prostate cancer group called the PPCG, and we are now analysing a cohort of about 1,400 prostate cancers. But today I'm just going to talk about the data from the UK, which was around 200 prostate cancers. So initially, we carried out whole genome sequencing on 92 primary prostate cancers, all from surgery, prostatectomy. We also included in our cohorts 20 samples from 20 metastatic patients, 10 of whom were hormone naive, and the other 10 had been treated with androgen deprivation therapies. They were castration-resistant metastases. Following whole genome sequencing, we carried out a set of analyses, including driver analysis, timing, heterogeneity, signatures, and kind of clinical outcomes. Okay, so this is the kind of the overall landscape that the genomic landscape that we observed in our 100 or so prostate cancers. You can see at the top the mutational signatures. These are the kind of the types of mutations that occur within the tumors and are indicative of environmental exposures to carcinogens. You can see the number of substitutions or single nucleotide variants in the second panel. The third and fourth panels show the number of small insertions and deletions, or indels, and the bottom panel shows the number of rearrangements or structural variants that they're often called. I'd just like to draw your attention to a couple of interesting things. So I don't know if you can see my cursor, but there's a set of boxes. It's the third row at the top, which show the sample type. And you can see that the samples that we have on the left-hand side are all these kind of brown boxes, which represent castration-resistant metastases. So these were samples that were taken from actually from autopsy from men that had died of prostate cancer. So they were very advanced castration-resistant tumors, and you can see that generally they had a large number of genomic aberrations. So they had a large number of SNVs, also a large number of indels, and a lot of structural variations. So generally, they were much more genomically unstable than the primary tumours. It's also noticeable, though, that if we look at the hormone naive metastases, which are the samples with the yellow boxes, they also tend to lie to the left of the figure, which means that they also had a large number of SNVs, and we saw that those samples also had a large number of indels and a large number of structural variants. So we don't think that this genomic instability was really driven by the drug treatment, the ADT. It's more that as tumors progress to metastasis, they generally do become more genomically unstable. They acquire more aberrations of various types. The last thing I'd like to point out on this slide is that a number of our samples had mutations in BRCA1 or BRCA2 or other genes associated with homologous repair defects. You'll notice that those samples which have BRCA defects, which are these green boxes at the top, which have got the red outline around them, also tended to have a particular mutational signature, which is this orange mutational signature, signature 3, and they also tended to be enriched for a particular type of indels. These light blue indels are microhomology mediated indels, which are associated with homologous repair defects. So we can use mutational signatures to identify samples that have homologous repair defects, and therefore may be more responsive to particular drug treatments or radiotherapy. So one of the aims of our study was to identify driver events. We used 3 different methods to identify drivers. The first was DN-DS analysis of SNVs and indels. So this looks at the ratio of non-synonymous to synonymous mutations, with the underlying thought that if we see enrichment for non-synonymous mutations, that's an indication of the driver status of a gene. However, we wanted to go beyond that. So because we had whole genome sequencing rather than whole exome sequencing, we were able to look at the whole genome, and we focused particularly on copy number events and structural variants, which wouldn't be obtained from whole exome sequencing. So we also identified regions of the genome that were enriched for either loss of heterozygosity, gains, copy number gains, or homozygous deletions. And we used a statistical permutation tests to identify these regions. The third method that we used was looking at the structural variants, and we compared our prostate cancer samples that were ETS positives, that had ETS fusions, with those that had that were ETS negatives, that didn't have ETS fusions. The reason for doing that is that we see quite a large variation in the rate of SVs. If we look across the genome, that's partly because of things like fragile sites. We have regions of the genome that are fragile, and so might be subject to increased rate of structural variation. So to kind of remove that confounder, we just looked within our set, comparing the ETS positive and ETS negative tumours. So this panel shows you kind of all of the genes that were identified as enriched within our data set. Again, I'm going to draw your attention to a couple of interesting things. So the second panel is a comparison. It highlights the genes that were found enriched in either ETS positive or ETS negative tumours. You'll notice that the two genes at the top, P10 and TP53, have a bar to the left, so they were enriched in ETS positive tumours, whereas the third gene, SPOP, was enriched in ETS negative tumours. So we can see already that that the two different subtypes of prostate cancer, which have been quite widely used for some years, actually have tend to acquire different driver genes, suggesting that maybe they're proceeding along different kind of evolutionary trajectories as they progress to cancer and onto metastases. So we also compared the primary and metastatic samples. We found that there were no genes which were enriched in primary rather than metastases, but we identified these 6 genes which were all had greater prevalence in the metastatic rather than the primary samples. And these genes were FOXA1, AR, P10, TP53, MYC, and NEAT1. Interesting that the bottom one, NEAT1, is actually a non-coding RNA. So that was a very interesting observation that was found enriched in prostate cancer, and specifically in metastatic prostate cancer. We noticed, looking at the individual driver genes, we noticed a few interesting things. I'm just going to show you a few of them. So in some genes, we identified hotspot mutations. This is just one example, CNOT3. So you can see the circles are missense mutations. The one triangle is an essential splice site mutation. But what's noticeable is that we had hotspots which had quite a high rate of mutation. So we had actually 5 samples which had mutations at one position, and another 5 that had mutations in another position, very close to the first position. All of these mutations caused amino acid change from glutamic acid to lysine. So we can see very strong evidence that we've got driving mutation here, causing an oncogene to be produced within these tumours in CNOT3. Second interesting gene I'll show you is FOXA1. So FOXA1 is it's been known for some years that FOXA1 is a driver in prostate cancer. What was particularly interesting to us here was that in addition to the coding mutations, which you can see the large kind of pile up in the middle of the figure, we also observed 6 samples that had mutations to the left, which were actually in the promoter region of FOXA1. This has not been seen previous to our paper, that you can also get enrichment for mutations in the promoter of FOXA1. Another method that we used, I mentioned earlier, is we looked at we compared ETS positive, you can see on the top, and ETS negative samples on the bottom. And we looked at both structural variants and copy number. And what was very interesting was that we saw that the kind of peaks in the distribution are different positions in the genome for ETS positive and ETS negative tumours, suggesting that selection has kind of operated in a different way on these two subtypes, and they're kind of being driven down different evolutionary trajectories. So in ETS positive tumours, we tended to see losses affecting FOXP1 and TBL1XR1, also a region on chromosome 5 which affects PDE4D, IL6ST, and MAP3K1, whereas in ETS negative tumours, we saw losses affecting different regions, affecting the genes ROBO1, ROBO2, and CHD1. Because we have evolution occurring all the time in cancer cells, that can lead to subclonal expansion. So this is where we have a subset of the cells within a tumour that have acquired particularly mutation, but they represent just a subset of the cells. So we observe heterogeneity within our tumour cells. This just shows you a few examples. So on each of these figures shows a different sample. All of them you can see have a peak at around 1, which represents mutations which are in all of the cancer cells, but they also all have a peak below 1. So typically at around 0.3 or 0.4. Which are the subclonal mutations, which are only found in a subset of the cells within the tumour. I'll just kind of zoom in on one example, which was in the middle of the screen and on the last slide. So this sample, PD14713, had 2 mutations in APC, both of which were just in a fraction of cells. We were very interested in this particular sample because it had 2 mutations in the same gene. On further exploration of this sample, we observed that it actually had a copy number loss, a deletion on chromosome 5, which is where APC is found. So we just had a single copy of chromosome 5 at that position. When we looked at the mutations, we could see that the 2 driver mutations were mutually exclusive. So when we look at the reads, which you can see at the top of the figure, we either had the mutation on the left or we had the mutation on the right. We never had them co-occurring. So this tells us the APC mutations were occurring in separate subpopulations of the cells within the tumour, and tells us that we had basically convergent evolution. So we had 2 different populations of cells which had both acquired a mutation in APC, which is quite interesting. So APC is a known driver in prostate cancer, but it's not anything like as prevalent as in tumour types such as colorectal. It's actually quite a rare driver in prostate cancer, but for some reason in this particular tumour, APC seems to be strongly selected for. So we don't know why, but it's apparent that in this particular tumour there was very strong selection for APC. So I've shown you a number of different kind of features of tumour evolution. I think I've only got a minute left, but I'm going to try and kind of quickly go through the rest of my slides. So what we wanted to do was put everything together from all the different aspects of tumour evolution. So we looked at all of the genomic aberrations that we saw within our samples. You can see listed on the left. We ran them through a neural network to divide them into subtypes, and we identified the 3 subtypes you can see in this figure. We also looked at structural variants close to AR binding sites. We identified 2 different groups, one of which on the left in blue was enriched for AR binding sites, and one was depleted for AR binding sites, you can see on the right. We also used a third method, which was to time the copy number events and driver events. So we identified early and late events within our tumours. And again, we grouped our tumours into 2 groups, which had undergone different evolutionary trajectories. Really surprisingly to us, when we combined these 3 different methods, we saw that the 3 different methods had actually identified the same grouping or very similar grouping of tumours. So we actually combined these methods, and we then found that one of our groups of tumours had much better outcomes than the other, with a hazard ratio of 2.3 between the two different groupings. We also were able to construct a stochastic model which actually modelled the evolution in stages as the tumours acquired different driving mutations, and we could see that they, starting on the left and moving to the right, tumours either moved to the top, which we call the canonical evotype, or to the bottom, which we call the alternative evotype, and they were driven by specific driver mutations. So, for example, you can see that if samples acquired an SPOT mutation, they would very rapidly move to the alternative evotype, whereas if they acquired something like a P10 mutation, they would slightly more slowly move to the canonical evotype. So we're able to construct evolutionary trajectories, which means we can actually predict the future behaviour of a tumour from an early stage, from the observation of driver mutations at an early stage. Obviously, there were lots of people involved in these studies, so I'm only able to show you a few people who are really central to this study. Yeah, I'd like to thank all of these people and the patients and their families who are involved in this study. And thank you for listening.
Thank you very much, David. So we also have time just for maybe one or two quick questions, if there are some burning ones now. So please. I see people being shy on Zoom, or maybe they keep the question for the end.
Hello.
Hi, I had a. Oh, Klaus, go for it. You were first.
No, I'm happy. Wait, please go ahead.
No, I just more than a question. I really like this APC story about those two subclones. I think it almost looks like a single clone given the frequencies in the population. It's two exclusive mutation in the same gene. Probably means they also occur at the same time because they had like more or less the same frequency. And I was just wondering, like, what can be the story? Like, do you have like two different cells acquiring an APC mutation at the same time, becoming like some sort of equal subclone? And I guess if you add the two frequencies, actually more than half of the population that get an APC mutation, whether one or the other.
Yeah. So the cancer cell fraction of those mutations was less than 50%. So from the kind of the maths of the CCF, it is possible for them to be either branching, so either be in separate subclones or for them to be in the same subclone. The only reason we were able to identify that they were branching, they were in separate subclones, was because they were actually very close together. So they were only separated by a few hundred base pairs. So we actually had read pairs that covered both mutations. And so we were able to see that they were, I think, mutually exclusive. But it was just lucky. So, I mean, I think it's actually quite likely that in other samples, we have a similar situation where we think we've only identified one subclone that is maybe in 40% of cells, but actually, in reality, there are two different subclones, but we're just not able to identify that. So it was just kind of lucky in this situation. I mean, we have carried out kind of larger studies within the PCOG consortium, where we had 2,700 samples across different tumor types. We've done a more extensive kind of study there where we've looked for these pairs of mutations which indicate branching subclones, and we have found that in quite a large proportion of samples which appear to have just one subclone, actually they do have two or more subclones that are branching. I mean, really what we need is more kind of multi-sampled studies where we sample different parts of the tumor, or single cell studies to really get at the heterogeneity.
Okay, thank you. For the sake of time, if you agree, Klaus, we keep your question for the end. I think we will have time. Okay, thank you. So thank you again, David, for the very nice presentation. I propose that we move on with the next speaker. So our next speaker is Professor Youqiong YE, who is a principal investigator at the Shanghai Institute of Immunology from the Shanghai University School of Medicine. And she will be talking about today the characterization of hypoxia-associated molecular features to aid hypoxia-targeted cancer therapy. So thank you, Professor Ye, and looking forward to hear your presentation.
Okay, thank you. Can you see my screen?
Yes.
Yes, okay. Thank you for the kind introduction. Today I will talk about the hypoxia-associated molecular feature. Because the professor which also have introduced like the ICGC is provide the multiple omics data resource. And in the tumor field, the TCGA, it started about the 2005 and close the 2018. And this database they provide the multiple omics data like the genomics, epigenomics, and like the transcriptomics. This data across more than 11,000 patients across the 33 cancer type. And also provide the clinical information like the age, stage, and subtype, all the smoking status or overall survival. Based on this, we can do many things about like the data mining. And besides this, the omics database, like the pharmacogenomics data database also have the important resource. For example, the GDSC, this database has the transcriptome data in about 1,000 cancer cell run. At the same time, this database, they also have the drug response in the more than 250 anti-cancer drugs. And these drugs are involved in the oncogenetic related pathway, like the PI3K signaling pathway or the RTK signaling pathway. So this database have the huge data, and also have the other database like the CTRP or CCLE. They also provide the omics data and the drug response data. But for this so many data, each omics data on its own, typically they provide a list of difference association with the disease. However, analyze only one data type is limited. So the multiple omics data and the multiple omics data are a complex regulate the network in the feedback loop. So the emerging challenge is how to integrate the data to elucidate the biological question and transfer this omics information into like the biomedicine and biological knowledge. So in this talk, I will introduce how to use this data across the multiple omics layers to experience our biological sense. So today I will focus on the hypoxia. Hypoxia is a condition that characterized by the limited oxygen level. From this figure, we can find from the left to the right, the oxygen supply was gradually decreased, and the tumor microenvironment changed from the normoxia to the hypoxia. And hypoxia is a feature of the most tumor, and it linked to the many cancer hallmarks that are associated with the metabolic reprogram, impair the genomic instability, or the impair the immune response, or increase the autophagy, or the apoptosis and reduce the proliferation. It also related to the treatment resistance, like as the chemotherapy, radiation therapy, or target therapy. And in our analysis, we also found the dynamic alteration of the hypoxia during the tumor genesis. From take an example for the lung cancer, we found a range from the stage 0 to 1 is we present the mucosa, the normal sample, to the stage 8 is represent the malignant cell. We can find that the hypoxia score is gradually increased during the lung cancer tumor genesis. In addition, we perform the spatial transcriptome by the fresh further tumor tissue section from the lung cancer, and based on the clustering and the spot feature, and we define the cell types among the spatial slide on the colon cancer, and we map then we map the hypoxia score on each spot, and we found that the malignant cell are surrounded by like the macrophagy and fiber they showed the highest hypoxia level. From this quantification, we can find that the malignant cell have the highest hypoxia status. We also obtained the spatial transcription from the melanoma from the previous study, and they also showed the consistent result that maybe the malignant cell will have the highest hypoxia score, while the other cell they maybe have a relatively lower hypoxia score. So in the tumor, the TCGA, they didn't detect the hypoxia status by the hypoxia sensing probe. So how to distinguish the hypoxia status in the large scale data set? So in here, to classify the hypoxia status, we employ the unsupervised hierarchical clustering based on the 5 filtering hypoxia gene signature. You can see for the top 3 subcluster were assigned into the hypoxia score high, hypoxia score intermediate, and hypoxia score low. So from the right panel, we can see the percentage of the hypoxia were divided into the score high, intermediate, and low. In these 21 cancer types, both these 3 groups have contained more than 13 samples. Then to assess the clinical outcome of our sample classification, we found that the hypoxia score tumor are associated higher the hazard ratio. Then they also associate, we can from this survival analysis, we can find this tumor are also associated with the prognosis in multiple cancer types. So this result suggests that potential prognostic power of our hypoxia status classification. Then to identify the hypoxia-associated molecular feature in cancer, we employed the propensity score to reduce the potential confound effect like the gender, stage, or the tumor state, or the tumor stage, age, or the tumor purity. Then alter the balance when we compare the molecular feature between the hypoxia score high and low to identify the hypoxia-associated molecular feature. So in this way, we identify then the significant difference between the hypoxia score high and low. Then the molecular feature, including the mRNA, microRNA protein, DNA methylation, somatic mutation, and copy number variation. And the fraction of the significant feature in the one cancer type is in one cancer types over the total feature in all cancer types, and the bar plot indicate the number of the significant feature for each omics. So the effect of the hypoxia state on the molecular data for different cancer types might vary significantly. For example, the mRNA expression alteration range from the hundreds in the ovarian cancer. However, they have more than 5,000 in like the TGCT, these cancer types. So this overall, this result was provide the overview, the molecular difference associated the hypoxia status across the cancer types. So to see we can each layer of the omics data, we can find like for the microRNA expression, we identify that the microRNA will also they showed the differential expression between hypoxia score high and low. Then we can find that like the micro 2010, they have significant upregulation in the hypoxia score high group in the 16 cancer types. Then they can, and this hypoxia-associated microRNA, they can regulate the mRNA, then they can regulate the pathway. So we can find that they can also slow the microRNA to regulate the pathway like the RAS signaling pathway or TGF-beta signaling pathway, HIF-alpha signaling pathway, and this microRNA expression are also correlated with the drug response. For example, for the micro-210, this microRNA, they can correlate with the drug for the positive correlation or the negative correlation. Take this example, we can see this the microRNA was negative correlation with the drug of the ATK inhibitor, and also the positive correlated with this is the microtubule this is the destabilizer. Then when use the then we follow the protein expression, and we use the functional RPA data to quantify the protein expression. Then we can find this RPA array, they cover the key cancer-related total or the phosphorylated protein. Then we identify the hypoxia protein alteration in the cancer signaling pathway, and the expression of this hypoxia. This protein may be associated drug, the sensitivity or the resistance. Then further, we review the global of the hypoxia-associated somatic mutation. Then we examine the mutation frequency across the multi-cancer type and identify hypoxia mutated gene may range than the 1 in the TH1M to the 14 in the LUAD. For example, like the tumor, the tumor protein, this is the TP53. They have the significantly high mutation in the breast cancer and the lung cancer, but they have the related lower mutation in the hypoxia score high tumor. Then the next layer is about the copy number alteration. We found that the hypoxia they can induce the transient site spatial copy number gain in tumor cell. So in our analysis, we identify the significant hypoxia-associated copy number variation in the 13 cancer types, range from the 2 in the TH1M to the A29 in the breast cancer. And take an example that for the this is the LGG, is the lower grade of the glioma. We can find this location have the higher amplification, then they might have like the BRAF or MET. Then this location, the amplification, that associates the drug response. To understand the effect of the hypoxia microenvironment on drug response, we have performed the integrated analysis to assess the association between the multiple omics hypoxia molecular feature to the response to the anti-cancer drugs in the TCGA patients. Take the ESCA as an example. We identify the 61 genes that are upregulated in the hypoxia score high samples. And these genes are hypomethylation and regulated by the 9 microRNA. And this expression of these genes are linked to the drug sensitivity response to the anti-cancer drugs, while the DNA methylation and targeted microRNA are linked to the drug resistance for the same anti-cancer drugs. And these drugs target important pathway like RTK signaling pathway or PI3K pathway. For example, this is the EGFR. We can find that the EGFR is a high expression in the hypoxia score high tumor in the ESCA, and the EGFR expression is linked to the drug sensitivity to these drugs, like this is the RTK signaling pathway inhibitor, crizotinib. Then we can find that this EGFR, they showed the hypomethylation in the promoter region. In the promoter region, then the DNA methylation, the DNA methylation of the EGFR is linked to the drug resistant response to the ATK inhibitor. And meanwhile, they have the 4 microRNA that target the EGFR. They showed a significant low expression in the hypoxia score high tumor, and these microRNA are positively correlated with the drug resistance to the corresponding drugs that negatively correlate with EGFR expression. So to further investigate the clinical amplification of the hypoxia molecular signature, we examine the molecular alteration between the hypoxia score high and low, and the hypoxia across this 5 layer of the molecular feature across the clinical optional genes that targeted by the FDA approved drugs. And these drugs are involved in the different type of therapy, like the immunotherapy, this is the homotherapy or chemotherapy and target therapy. And then we found that about 90% of the clinical optional genes are associated with at least one type of the hypoxia-associated molecular signature in at least one cancer types. And this different shape was present the different molecular signature associated with the hypoxia. Then we further to find the hypoxia effect on the clinical optional gene. They can link to the hypoxia effect response directly. For this bar was showed about the 21 FDA-approved anti-cancer drugs are available in the GDSC database. Then we showed the number of the significant correlation of the hypoxia score and the drug response across the multiple cancer types. And they showed that the hypoxia associated with the drug resistance and the drug sensitivity response to the anti-cancer drugs. Then to directly validate our finding on the drug response, we perform the drug sensitivity experiment on the selected drugs, like under the hypoxia condition, about the 1% of hypoxia. Then the normalcy condition is the 21% of the oxygen in the two lung cancer cell lines. Then consistent with our computational prediction that these two drugs, that about the top one inhibitor, the Genomic receptor antigen, they showed the greater the drug resistance under the hypoxia condition, while the drugs, the AKT inhibitor or the RTK signaling pathway inhibitor, they showed the great sensitivity under the hypoxia condition in both these two cancer cell lines. So this result showed that the hypoxia is largely affect the drug resistance and the drug sensitivity response on the anti-cancer drugs. However, in the traditional concept, they always said that hypoxia is associated with the drug resistance. But in our study, we also found that sometimes hypoxia they may have associated with the drug sensitivity response. So take home message that in our study, we classify the hypoxia status across the TCGA samples and identify the multi-omics hypoxia-associated molecular feature and link this association between the hypoxia status and the drug response. And we also found the hypoxia have the therapeutic liability. So we saw when in the following clinical trials, maybe we needed to consider the hypoxia status of the patient. Then some patient with the hypoxia score they maybe benefit with some drugs. Okay, that's all. This is my presentation. Thank you.
Thank you very much for the presentation. We have again time maybe for just one short question. So please, if anyone wants to ask something. Now, maybe a very short one, quite naive because I'm not really working on this field, but how much can the hypoxia score that you use separate really hypoxia from tumor proliferation? Because I saw, for example, that you have association with TP53, certainly associated also with proliferation in lung cancer, for example. So how much what you see is really hypoxia and not also tumor proliferation?
You said the tumors. What's the tumor? Can you repeat your question? It's about the hypoxia for
hypoxia and tumor proliferation. So because the two are certainly linked, and how much what you observe, the correlation that you observe is really hypoxia and not also tumor proliferation.
Because this because we just have the for the hypoxia, we only consider that we balance the other confounded effects, and so sometimes we can find the very significant feature in some cancer types. So only just one or two they can pick up for the significant under the significant control.
Okay. Thank you again. So I propose that we move on with to the next presentation. So the next speaker here is Dr. Claus Jorgensen. So he is a senior group leader at the Cancer Research UK Manchester Institute. And I don't know yet the topic because it was returned to be confirmed in the program that I have. So I will let you, Claus, tell us about your topic.
Thank you very much.
Thank you.
Let's just see if we can get this to work. All right, thank you. Can you see the presentation?
Yes, we do.
All right, okay, perfect. So thank you very much for the very kind invitation to talk a little bit about the stuff that's going on in my lab today. So the title of my talk is the tumor microenvironment, it takes a village. And essentially what I'll go through is to talk a little bit about kind of the other aspect of solid tumors in particular, which is kind of focusing on the microenvironment and how the microenvironment regulates tumor cell function. And so this is really illustrated quite well in a solid tumor such as pancreatic cancer, which on average 80–85% of the entire tumor volume is actually non-tumor cells. And so the first slide we can see here on the screen is actually, it's a slide from a resected patient pancreatic cancer, where the tumor cells or the epithelial cells are stained with cytokeratin in green, and kind of the magenta cells are vimentin positive stromal cells, really illustrating the abundance of the tumor microenvironment in these tumors. And so this is also quite well preserved when you look down the microscope. In this particular case, it's a pancreatic cancer from one of the animal models of pancreatic cancer that's driven by oncogenic KRAS together with a point mutation in P53. And again, when you're looking down on these tumors, you can see that the tumor cells here outlined in black are kind of dispersed within this microenvironment, which is outlined in red. And when you then start staining for various markers of cells and proteins in the microenvironment, it becomes apparent that we have a very kind of immune myeloid-rich microenvironment with very few T cells sustained by CD3, but also we got an expansion of cancer-associated fibroblast stains by alpha-S-smooth-lactin and a really highly abundant remodeled extracellular matrix such as stained by Picosorius red and senescent C. And so this is really a constant evolutionary process, and I think probably some of the best data we have of that actually originates from studying some of the preclinical genetic engineered mouse models of pancreas cancer. And so these are typically driven by tissue-specific expression of oncogenic activating mutations in KRAS, which in patients is very common. It's more than 95% of all patients that have activating mutations in KRAS. And then together with loss of CDKN2A, P53, or SMAD4 function, which are the most frequent inactivated tumor suppressors in pancreatic cancer. And so when you study the microenvironment in these animal models, what becomes clear is as even at these early pre-malignant stages, the pancreatic intraepithelial neoplasia, you already have a really abundant infiltrate of monocytes, macrophages, neutrophils, and we see an expansion of the cancer-associated fibroblasts, but that the CD8 T cells remain very poorly infiltrated in these tumors. And alongside these cellular changes, there are extensive changes in the gross environment, such as biophysical changes, where we have an increase in the tissue stiffness, we have an increase in the intertumoral pressure, and as a consequence, as a decrease in the perfusion rate, which have been hypothesized to lead to hypoxia, but also poor delivery of therapies in this hard to treat cancer. So one of the things that's kind of interesting in terms of understanding the impact of the tumor microenvironment is to try and look at some of the data of these animal models of pancreatic cancer. And so if you express only activated forms of KRAS in the pancreas in animal models, most of the animals develop these precursor lesions, pancreatic intraepithelial neoplasia, and very infrequently they proceed to develop a full-blown adenocarcinoma. But if you delete P53 or inactivate P53, a much higher fraction of these animals go on to develop full-blown adenocarcinoma, and that's really a well reflection of the genetics we see in patients as well. Now, the driver commonly used in these animal models is actually expressed at very early stages during animal development. And so you have the induction of oncogenic KRAS and loss of P53 at very early stages of tissue development in these animal models. Now, instead, if you do the same experiment with an inducible CRE that is only expressed in the adult tissue, what happens is when you do the same genetic that in young animals leads to overt pancreatic cancer, in the adult animals, you still only see pancreatic intraepithelial neoplasia. But if you induce inflammation such as by cerulein, you actually have a full-blown adenocarcinoma. And so that kind of indicates that actually normal tissue restrictive factors are in place that prevent the transition between PANIN to full-blown adenocarcinoma. And when we need to remodel the tumor microenvironment for tumors to progress. And based on this data and much other data in the field, people have really started to untangle these sort of interactions between the tumor cells, the cancer-associated fibroblasts, T cell exclusion, and myeloid cells, as well as the remodeling the extracellular matrix, and several therapeutic strategies have been put in place to kind of test these hypotheses. And fair to say that while several of these approaches have looked very promising in preclinical models, they have still to make an impact in patients in the clinic. And so there could possibly be many reasons for this. A couple of reasons could be that the way we understand these interactions are not sufficiently fine-grained enough to actually decide which patients to get which treatment. And the other part is that we still rely very much on animal models to study these tumor and stroma interactions, and therefore it's still very difficult to really assess them preclinically in patient-derived models. And so it kind of gets to the key question of the emphasis in my lab is, how do we really untangle these complex cellular interactions between tumor cells and cells in the microenvironment? And our approach has really been, sort of inspired by a rough engineering type of approach to things that, if you want to understand how a bicycle is working, the best approach is to take it apart and put it back together again. And then you can start to understand how individual elements are needed to interact for the bicycle to work. And so for the purpose of this talk, I'll focus on kind of our two approaches to illustrate this, one of which is recent study where we've started to map out what are the cellular constituents of these tumors, and the other is development of some models where we can start to understand and model in a patient-derived manner how tumor cells interact with cells on the microenvironment. And so for the purpose of the first part of the talk, to map out the constituents of the cellular elements in these tumors, we took a single cell approach, and rather than doing single cell RNA sequencing, we went on and did a labeling antibody-based approach. And so for this particular approach, we used mass cytometry or cytometry by time of flight. And it's really, if you will, technically or from a principle perspective, it's really rather simple to your general FACS approach, where you have a single cell suspension of cells, you label them with antibodies, but actually rather than having fluorescent labeled antibodies, you use a metal tag on the antibodies. And you then pass them through a front end on an instrument that's really a microfluidics device. And then you introduce them to ICP mass spectrometry. And so an ICP MS is kind of a well-established technology to measure metals or rare earth metals, kind of been used a lot for elemental analysis. And what happens is that all organic material is first completely disintegrated in the torch, and then the ensuing metals, which are now ionized, are read out in a relatively simple time-of-flight mass spectrometry. And what that means is that because of these rare earth metals have very little spectral overlap, you can actually generate really high multiplex analysis approaches, analyzing 40 to 50 antibodies at a single cell level. You study protein levels rather than mRNA, and you get a relative quantitative piece of information about the expression level of these particular proteins of interest. And so we developed antibody panels and developed approaches to study this using, again, in this particular case, tumors isolated from the animal models of pancreatic cancer, where we isolate the tumor, we dissociate it, we then stain and barcode, meaning that we can analyze multiple samples simultaneously by CyTOF. And then we studied the immune system, so both the innate, the adaptive, and the mesenchymal cancer-associated fibroblasts. And so the real advantage of these approaches is really just the number of cellular events you can read out. So from 19 tumors we analyzed in this approach, we get 5 million single cells read out with 40 plus markers. And as you can see here, just to illustrate, overlapping with CD45 expression, it kind of gives us an idea of the abundance of the immune infiltrate compared to non-immune cells in these tumors. And so just by way of illustrating what we can do with these sort of data, if we then focus in on mesenchymal stromal cells and do a non-supervised hierarchical clustering, we can start to identify individual subsets by the expression of individual markers. And I should just mention here that a lot of the markers we've chosen for this particular panel are really cell surface markers, because we acknowledge the fact that one pitfall with a lot of the approaches in terms of studying the tumor microenvironment is that we're currently very poorly equipped to isolate the individual stromal cell populations and study them functionally. And so we kind of need to go from just a pure mapping effort to actually a functional annotation of individual subsets. So by studying this sort of map of individual stromal subsets, we can start annotating the mesenchymal subsets. So for example, by the expression pattern of CD31, MCAM, and other markers, we can annotate individual subsets to blood endothelial, lymphatic endothelial, and pericytes. And then we can start analyzing the remaining mesenchymal stromal cells, which are fibroblasts or cancer-associated fibroblasts in this particular case. by expression of common markers, we can see that we have, you know, a lot of the typically used markers used to annotate cancer-associated fibroblasts are really expressed in non-overlapping gradients, suggesting that these annotate subsets, and actually, you know, taken together, this also illustrates that we don't have one particular marker that's suitable to annotate all cancer-associated fibroblasts without any overlap with other cell populations in the microenvironment. One particular marker we got particularly interested in was CD105, which is a TGF-beta co-receptor, and this is in particular because it's expressed in an almost binary manner. So we have CD105 negative cancer-associated fibroblasts and CD105 positive cancer-associated fibroblasts. Going into human cancers and doing multiplex IF to see if CD105 positive or negative cancer-associated fibroblasts were also present in human pancreatic adenocarcinoma. We can then, when we overlay using Vimentin as a general marker for the stroma with CD105, we can see that we have CD105 positive, Vimentin positive areas, and we have Vimentin positive CD105 negative areas in the human cancer, suggesting that indeed these two subsets are also present in human cancers. To annotate the function of these individual subsets, we set up in vivo experiments where we co-injected tumor cells with CD105 positive or negative pancreatic fibroblasts subcutaneously, and then we could use that to annotate the tumor growth as well as the function of the fibroblasts. And this experiment here is just really the control experiment to demonstrate that the CD105 positive fibroblasts remain positive and the CD105 negative fibroblasts remain negative when they're co-injected with tumor cells. And we can identify them locally 7 days after co-injection, but actually in fully developed tumors, we don't see many fibroblasts, suggesting that actually the effect, which I'm about to show you, it probably happens early during tumor growth. And so this was for us where it got really interesting. So what you see here in the gray bar is the tumor cells injected subcutaneously by themselves. So this is essentially what the tumor cells grow like by themselves. If we co-inject with CD105 positive fibroblasts, you can see that there's a slight acceleration of tumor growth, although it is non-significant. But what's really interesting is if you co-inject with the CD105 negative fibroblasts, you see a tumor suppression, and actually in about 40% of these tumors, we see complete rejection of tumor growth. This is dependent on a functional adaptive immune system. So if we do the same experiment, but we inject the tumor cells together with fibroblasts in animal models that are completely deficient for a functional immune system, we can see that both the CD105 positive and negative co-injection of fibroblasts kind of really tracks very nicely with the tumor cells injected by themselves in gray again. If we use RAC1 animals which are deficient in B and T cells, we'd see the same phenomenon. And if we repeat the experiment in animal models that are deficient for BAC3, which have deficiency in CD1, so dendritic cell maturation, we see also a slight loss of the tumor suppressive effect of the CD105 negative fibroblasts, suggesting that the effect of the CD105 negative fibroblasts in terms of repression of tumor growth is dependent on a functional adaptive immune system. And so to summarize this first part of the talk is, we've kind of started picking away and have been able to identify that, that there's a complex interaction between tumor permissive and restrictive elements of these tumors, and therefore suggesting that our approaches to targeting the microenvironment needs to identify and target promoting signals while possibly augmenting the tumor restrictive signals. And so that kind of gets me to the second part of the talk, because, you know, how do we start considering testing the effect of the microenvironment, in particular, both the biophysical and the cellular changes in patient-derived models, rather than relying so much on animal models. And I think it's fair to say that in particular, you know, organoids as a preclinical model have really fundamentally changed how we can do these sort of studies now. So, as you're probably very aware, that organoids are stem cell like cells that you can grow from humans in the laboratory, and you can get them from both normal tissue and from tumor tissues, and allowing you to kind of study these cells, and they actually really preserve particular features of the tumor cells, and you can expand them and grow them for extended periods in the lab. Now, when you're looking down the microscope of these organoids that are grown in 3D in matrigel, you get these beautiful cells grown as bubbles. But what we really need to kind of take these models closer to what the tumor actually look like is we need to be able to think about how we incorporate cells of the microenvironment. And so to do this, we teamed up with Linda Griffith, who is a bioengineer at MIT, who have this fully synthetic hydrogel system for cell growth. And the way it works is you have these PEC 8-armed molecules you can attach various peptides to in a modular manner, allowing us to mimic specific adhesive cues for the cells to adhere to. You can then cross-link it, and you can then start embedding organoids and other cells. And so we did a lot of upfront work to identify the best combination of these sort of peptides and the features of the PEC gel that I, for the argument for the sake of time, won't have time to go through today. But suffice to say is when we then test these synthetic hydrogels, we can actually grow both urine, but also importantly, patient-derived organoids in these completely synthetic scaffolds. And we can, by staining for markers and other features, we can see that the organoids recapitulate the same features that we will see when we grow them in the cellular derived matrigel. We can mimic the stiffness changes, which you can't do with matrigel. So these tumors, as I said earlier, are really tremendously stiff. So when you measure the stiffness across both animal and human samples, you can see that the median stiffness as measured by AFM is actually 3.5 kilopascal, whereas the normal tissue is about 1.5, and actually you get up to 20 times the stiffening of these tumors very locally. And we can recapitulate this entire stiffness range using these synthetic hydrogels, starting to consider how this might affect tumor cell function and tumor growth in response to therapy. Equally important, we can incorporate stromal cells such as macrophages and fibroblasts. So we have developed co-culture models where we can grow the organoids together with these stromal cells for extended periods. If we analyze the stromal fibroblasts by mass cytometry, we see that they adapt similar features as we would when we studied the tumors coming out of in vivo. And what we can also start doing is we can start studying using live cell imaging, just how the tumor cells here in purple remodel and how there are changes in the behavior of the stromal cells close to the tumor cells. And so to summarize this, we think that this is possibly a step change, allowing us to essentially study human derived models in a more contextual relevant setting in terms of understanding the biology, but also the therapy and therapeutic response. And just, you know, with the thinking about the wider challenges with studying the tumor microenvironment, kind of opening up for discussion, I think some of the things that are very worthwhile considering is, you know, how if we are thinking about targeting the tumor microenvironment clinically, I think it's absolutely critical that we continue to characterize the tumor and the stroma interactions in patient-derived models, but also really start thinking about how we can analyze the tumor microenvironment in a clinical setting, because still, you know, we rely heavily on tissue for these sort of analysis, and for in a lot of cases, it's still very difficult to actually get the intact tissue from patients to derive these sort of key observations for patient stratification. And so with that, I really want to acknowledge everyone in the lab, and also the very kind support from our key funders, and you for your attention. I'm happy to take any questions. Thank you.
Thank you very much, Claus, for the fascinating presentation and this kind of futuristic organoid that you showed us. We have time for maybe one very short question, if there is one burning. But we are also approaching anyway the final discussion. So maybe we keep the questions for the final discussion and question answer slot at the end. So we can almost, yeah. Okay, thank you again, Claus. So we move with the next presentation from Dr. Benjamin LEMASSON. So Benjamin is a researcher at the Grenoble Institute of Neurosciences, and he's going to talk today about segmentation and classification of glioblastomas using AI-based tools. So Benjamin, please.
Hi, thank you for your introduction. So yeah, so I'm working in the Grenoble Institute of Neurosciences, so it could be far from cancer, but actually we are, I'm in the team of neuroimaging and brain perfusion. So we are working on brain disease, and among others, we are working on glioblastomas. And in the lab, I'm working more on imaging biomarkers. So and for the brain tumor, I'm working with colleagues in Toulouse, so in Oncopole. So today I will introduce you to the techniques we are developing. So we are methodologists, and we are developing new approaches to exploit and extract information from neuroimages. So first of all, I would like to introduce just what we call artificial intelligence, because for a few years, everybody is talking about artificial intelligence, but actually it's a really old concept. First paper came out 70 years ago, but in the artificial intelligence, it's a big bag, and inside where you have machine learning approaches, where you can find family word like a random forest technique, regressions, and super vector machines. But more recently, new approaches arrived, like neural networks, and even really 10 years ago, we have a deep learning approaches. So why now are we talking about artificial intelligence, and especially in the field of images? It's due to three main developments, which is the hardware. The hardware are now strong enough to exploit and to analyze using deep learning approaches. You also have software developments, mainly pushed by Google and Facebook in America. And also you need the last part is the big data. And for images, it's pretty new to have large databases, structured databases, like a cancer imaging archive. And altogether, this technique proves that the deep learning approaches, so the most recent one, has a strong impact on the development, and the algorithm developed using such techniques are really outperform the oldest approaches. So it will depend on the data available, and also on the performance. The performances will depend on the data available. So today I will present you several developments we are doing using machine learning and deep learning approaches. So just one last slide for the introduction is the difference between the machine learning and the deep learning. So here's an example with a car, a simple task where you want to automatically detect if the picture contains a car or a train or a cat, something else. So for using machine learning approach, the developer, so the engineer or the researcher have to develop himself or herself the feature to extract. Let's say for a car has four wheels. So wheels are on the floor. There is some windows. So you have to develop all this. You have to develop and to code all these features you want to extract from the image, and then you are using normal and classical classifier such as random forest, and at the end you will train your model and you try to find if the car is present on the image or not. When you are using deep learning approaches, you will get rid of the feature extraction. So the network by itself will learn which feature is important to classify this picture. So the deep learning is really nice, but to do that, you will need a lot of examples, not a lot of data. And like I said, you will need a lot of computer power and a good software to handle everything. So today I will present you some development on machine learning and deep learning applied to new imaging biomarkers. So this is this is the classical main steps when you want to exploit images in the medical field. So you have raw data, so data just collected from the clinics. So here it's an example of a human brain tumor. So this is one tumor, but with multiple contrasts acquired by MRI. So you have the T1 gadolinium on the top and the FLAIR imaging on the bottom. And then if you want to characterize or to link these images to an outcome, which is maybe the patient outcome, survival, and so on, you have to quantify these images. So the first step you need to segment the lesions. So it could be manually, but of course it's time consuming and it will take forever. So we are using machine learning or deep learning approaches to do it automatically. And after, from there, you will extract information. So today we talked already about genetics, genomics, proteomics, but also in imaging we have radiomics. So the radiomics approach is a new term. Like 10 years ago, we introduced the radiomics. Is the fact to extract quantitative information from an image automatically and robustly. So here, if you have two images with two regions of interest, you can extract, of course, the tumor size, but also some information about the shape, about the texture, about the wavelength, and a lot of metrics, mathematical metrics to quantify these images. So you can easily acquire and extract hundreds of metrics from one image and one region of interest. But of course, you can mix things and put the one region of interest on another contrast, and so extract other metrics. So at the end of the day, for one patient, you will you can easily have thousands of metrics, and these metrics will be used to predict the overall survival, the time to progression, or anything you want to predict in the clinical point of view. And the last thing, we try to introduce clinical data to this methodology, to like age, or it could be genetics or proteomics or immunomonitoring metrics. So this is the typical main steps to develop new imaging biomarkers. So today I will present you three, maybe two, if I don't have enough time, techniques we developed. So using deep learning, machine learning, with the goal to improve the segmentation or the detection of different outcomes. So the first is the deep learning segmentation. So in this study, we have a lot of ready-to-use tools developed from maybe 10 years ago to segment glioblastoma, but most of them will focus on pre-operative glioblastoma. So like a lot of people say, is people bearing brain tumors will go to surgery, and at one point, so you will have a cavity post-surgery will appear on the scan, on the images, and we don't have right now any deep learning tool, any automatic segmentation tools that will handle this cavity. So we know why, because there is a lot of large data sets for preoperative glioblastoma, but there is no public postoperative data set large enough to carry a deep learning algorithm. So we wanted to evaluate a new way to use deep learning, which is called the transfer learning approach, with the goal to segment the postoperative glioblastomas. So what is transfer learning? So you have two tasks. The first task is you have your source task and the target task. The source task is where you have a lot of data and you can train a model on using a lot of data, and you will here in this example we use a large data set manually segmented by a lot of radiologists, so almost 500 cases, and from these 500 cases we could train a model, a deep learning model, to segment the preoperative glioblastomas, and the preoperative glioblastoma will segment the edema and the tumor core. And once our model is trained, we can transfer the knowledge to a small, tiny cohort we have. It's a post-operative cohort with a cavity. It's only 17 patients, so it's not enough to train the deep learning models. But using this transfer learning, we can do it, because we will transfer the knowledge from the previous learning to the new learning. And in this second task, we want to segment the tumor edema, but we also want to segment the cavity. So this study has been done using data from Toulouse. I will not go into the details here, but we use a general network used by the community, which is UNET. And we just modify the network, because at the beginning we have 4 images, MRI images, and we segmented 2 lesions. And after we use the same network, but we want to segment another lesion, so 3 lesions at the end. So what we found here, it's interesting because we use the metric used for such study it's a DICE metric. It's just a similarity matrix. What you have to understand, if the DICE is equal to 1, that means the automatic segmentation is equal to the manual segmentation, so it's perfect. So if we learn a deep learning model using only 17 patients, of course, you have a bad result, because it's almost equal to 0 for the cavity, for instance. The edema is large enough, so you can segment edema quite well. But if we use the transfer learning approach, so you pre-train your network using almost 500 patients without any gravity, but after you fine-tune your model using your small cohorts, you can increase every DICE. So it's quite interesting. This approach is quite interesting to fine-tune and to refine a model on a really tiny cohort you can have on a dedicated task. So this study was an example of using deep learning to improve the segmentation. I have another example here. It's more machine learning approach, and here we wanted to detect the radionecrosis. So why we want to detect radionecrosis? So when you treat patients with brain tumor and and with brain tumor using radiotherapies or immunotherapies, you will have a different tumor responses. Of course, everybody hope to have a true response, but it's not always the case. Sometimes you have a tumor progressions, and sometimes we have what we call radiation necrosis. The problem is to distinguish the two last. The tumor progression versus radiation necrosis is a difficult task. And this small example here, you have one tumor on the bottom and another one patient on the bottom, another one on the top. And the patient on the bottom is both are under therapies, but based on the image, it's difficult to say which one is responding and which one is in progression, because the radionecrosis look alike the tumor progressions. So we wanted to develop an algorithm to automatically detect that. And because we are using a multi-center data for this this study, we also had to face what we call the scanner effect. So this is a bias induced by a multicentric trials. So I think in genomics, you have the same issue when you collect and you pull data from different databases. You have some effects induced by the machine, and sometimes it's hard to pull the data without doing anything. You have some bias due to the acquisition. So in this study, we try to answer our question, and also we try to use new techniques to remove the bias induced by the scanner effect. So the cohort here, we use a multi-center cohort led by Toulouse Oncopole. So it's phase 1-2 glioblastoma patients treated by radiation therapy, or a combination of radiation therapy and immunotherapy. So the study is still ongoing. So for the imaging part, we are using a subcohort of 28 patients. Follow over time, so it's 122 MRI examination. Each data set included 6 MR scans, so it's a 3D images of patients, and the data come from 6 centers in France. So here you have the overall data analysis. So in imaging, you have a lot of pre-processing. So from the raw data here on the top left, and at the end, you want to do your classification. Is there any radionecrosis or not? You have a big pipelines in between. I will not go into the details here, but at one point you have the segmentation. So in this study, we use manual segmentation because the cohort was not really big. And from this segmentation, we extracted what I said, the radiomics metrics. So the radiomics could be a first-order metrics, but also could be texture metrics or shape metrics from the region of interest. After we had some trouble with undersampling, so in our cohorts there were only a few, I would say, less people with radionecrosis than true progressions. So we have to use statistic techniques to compensate. After we have our multi-center effect bias, we have to compensate too. And at the end, we built our model, our radiomics model, using a bootstrap approach and feature selection and classification. Here it's for the scanner effect. So we have a scanner effect. Here you have just a tiny example of 3 scanners, so 3 acquisition made in 3 centers in France. So using a region of interest, which is a healthy region of interest. So the histogram should be the same, basically. But we can see that the histogram on the left are really not the same. And on the right, we use a combat. It's an algorithm developed at the beginning for genetics and genetics purposes, and is now used in the imaging field. And the goal here was just to make the histogram look more alike from each other. So here we can see that the harmonization is working. And this is another representation of this effect. So at the beginning, before harmonization, you can clearly see that you can recognize 3 centers. So the metrics should be mixed, and but it's not the fact. You have a bias coming from the acquisition. And after harmonization, you cannot distinguish any scanner, any center. So it's nice. You can harmonize your data, your metrics. But the next question was, is there any impact on the prediction? Because our clinical question is still the same. We want to predict the radionecrosis, not to predict. We want to detect the radionecrosis. So it's what we have done here. We built several models. So we had 6 MR parameters. It's 6 MR scanner, so 6 images, and we can build we built detection models using any of these MR scanners, and after we tried to mix the metrics, and we did it with and without harmonization. And here we found that almost in every cases the harmonization improves the performance of our models, so we can more accurately detect the radionecrosis. And also another important result in our field is the perfusion images alone is enough to detect the radionecrosis. So this was an example of how we can use machine learning to do clinical tasks to help radiologists and medical doctors. And if I have a few more minutes, I just added this example here where we can use machine learning to mix segmentation and classification in one tool. So we did this study in preclinical this time. So we have animal models. So I will just keep this part because I think everybody is aware that the tumor is heterogeneous. So here it's in MRI we have a lot of data. We can acquire what we call multi-parametric images. So it's on your left it's animal models. So it's brain tumor models implanted in rats, and on your right it's human data. And you can easily follow your patient or your animals over time, and at each time point you can acquire a lot of data. So here you have oxygenation map, blood volume, blood vessel size, vessel permeability, water diffusion. So it's a lot of information at each time point. And the goal is to extract quantitative interesting information here to answer is it possible to automatically segment the tumor and classify the tumor using this data. So here we use a machine learning and a cluster analysis approach. So on the left, we have healthy animals, so healthy brain rats, and we built a cluster analysis just to define what is the normality. So it's an automatic cluster analysis. So it will just combine and give you an answer and a classification. And even if there is no spatial information in the cluster analysis, we can clearly see that the result is very nice. You have the cortex and the corpus callosum and the ventricle well defined based on the result of the classification. And after, the next step was to use and to apply this LC cluster analysis on pathological data. And it's what we did. And this application was, when we apply the normal models on pathological data, it's really easy to highlight abnormal voxels. So every voxel which are not defined in the cluster analysis from LC data were selected and automatically selected when we did this step. And when we compare versus the manual segmentation, it's almost perfect. So here there is no there is no a priori. I mean, we just learn a model from LC brain, and we apply it to pathological brain to detect outliers. So we did it for 4 glioblastoma models, so 9L, C6, F98, RG2. And we obtain a really good dice score compared to manual segmentation. So you have the automatic delineation in red, and on the last line you have the manual segmentation. So it's almost it's really nice because it's fully automatic. And in the last step, we try to say, okay, now we have the segmentation of the lesion. Is it possible to characterize the lesion using the same approach? So we did the second classification, a second cluster analysis, but this time just using the abnormal voxels. And this is a result. And what is really interesting is we can have a glioma signature, so automatically for each glioblastoma. And from there, we did it on a lot of data. And this is during a learning process. So we did a machine learning, we built a machine learning model to learn the signature from each models. And after we could define the abnormal fingerprint models for each glioblastoma. And we evaluated on a second data set. And here we demonstrated that we could be able to automatically segment and detect two kind of glioblastoma models, the 9L and the F98, with a really good accuracy. So to conclude, we are trying to transpose this technique in clinics. So this is just a few examples of data where we apply the cluster analysis. Here, which is really nice by this approach, is we have the interpretability. So when we use deep learning approach, it's really hard to have interpretability. You have some results. Sometimes you do a good diagnosis, but the clinicians always ask why it's working. So here, using this cluster analysis, when we have the signature of the glioblastoma, we always have the interpretation. So why this signature is composed of 5 classes, and this classes is composed of this compound of T1 gadolinium, diffusion map, flare map, and so on. So we have the interpretability. And the second development is for tumor longitudinal follow-up. So here it's on preclinical side. We apply this technique to monitor the glioblastoma growth. And here you can see on this tiny small example, it's we implanted the rat with the same glioblastoma lines, so it should be the same growth rate. But we can see that in the 7-day, 9-day to 17 days post implantation, and you can see that one glioblastoma the growth rate is higher than the second one, and we also can see that the tumor signature, so the heterogeneity is not the same, and the tumor signature is not the same. So this could be a good imaging biomarker to really diagnose the aggressiveness of the glioblastoma. So to conclude, the take-home message of my presentation, it's you have new AI-based tools which arrive for image analysis. And just because of the, like I said, of the hardware, software, and databases, that will really move forward and revolutionize the imaging biomarker field. And this will lead to more personalized medicines, because we hope to arrive in clinics and to analyze data in real times, and this data analysis will help the clinicians. But of course, such tools will need more structured and labeled pathological and LC cohorts, and we don't have enough data yet. And also strong validation using multi-center cohorts, and therefore we need to manage a multi-center bias. And for the deep learning algorithm, didn't have time to develop here, but we need to integrate more explainability tools to help the tools to go to clinics. And I would like to thank, so this is my team in Grenoble, and some of them are working especially on this project. Thank you.
Thank you very much, Benjamin. So we are still on time for having a short question and answer session. We have 10 good minutes. So what I propose is that we start with questions in particular for Benjamin, because that was the last presentation, and then we open up for questions for any presentation or more general discussions maybe. You can raise your hand if you want me to just give you the opportunity to talk, or just talk if no one is talking also.
I can just ask a question. It's just, so in the first project you presented, you have the transfer learning. So basically you train your model to recognize edema or tumors, and then you transfer to recognize something that hasn't been trained to recognize, but because it has like similar features, maybe it's just a round shape, it can actually recognize it.
Yeah,
because it's been trained to recognize something similar.
Yes, so the transfer learning is also used when you train your model using like picture, which is cars and birds and so on. People train their network, because the network is composed of hundreds of millions of parameters to tune. So sometimes they try to pre-train the models on large databases, and after the models start to learn how to recognize features, because at the beginning you don't know anything, it will learn by itself. So it try to learn to recognize round features, shape features, and so on. So yes, this principle of transfer learning is to pre-train and to adapt more easily the model. Don't start from scratch, but from a pre-trained models. So you can do it from 2D images from the life, but most of the time we try to do it with medical images.
Okay, but there's still retraining.
Yes, there is what we call fine tuning.
Okay.
So just the fine tuning, so the network, you can we train everything, or you can say, okay, for the encoding part, we know the model is already well trained, so we just will fine tune just the last part.
Thank you. We have Claus, who didn't have the opportunity to ask his question earlier. So please, Claus, and sorry for that.
Thank you. Thank you. So actually, I have a question first for David WEDGE, and then later on one for Pierre. So David, if I understood you right, so you're saying that by understanding or measuring early mutations, you can kind of predict, future trajectory in your models, right? So my question is, so in terms of those early mutations and how they arise, do you think that's just a stochastic event, or do you think there are other factors that might influence why a particular tumor acquired that mutation and have that particular trajectory?
Yeah. So I think, the study that I presented today was just entirely focused on prostate cancer. In prostate cancer, we seem to have a lot of complexity. So there are a lot of different mutations that can drive prostate cancer, and we identify a large variation in the trajectories across the different tumor types. I think if you looked in something like colorectal cancer, or in some of the blood cancers, then you might see a kind of narrower range of possible trajectories. I think those cancers appear to be a bit more kind of deterministic, and there's more certainty about the evolutionary trajectories. Within prostate cancer, yeah, it does appear that there's very large variation in the possible drivers, and they can be acquired in various orders. So we've had to construct quite a complex model for prostate cancer, so we don't actually identify any genes as being essential to driving any individual tumor down a particular evolutionary trajectory, but we can identify probabilities that acquiring a specific aberration will drive a tumor down one trajectory or another. So it is quite a complex kind of stochastic model.
Oh, that's interesting. So I guess as a follow-up question, why prostate cancer is so different than other sort of cancers? Is, has that something to do with inherent plasticity in the lineage and that gives rise to the tumor cells? Or is it something else, you think?
Yeah, I don't know. I mean, actually, prostate cancer, as we know, is actually very, very common. If you look in the prostate of men over a certain age, then it's very likely they will have prostate cancer. But the majority of prostate cancers tend to be fairly benign. Only about 15% of them will actually metastasize and become aggressive. I mean, I don't know, but the prostate is quite a self-contained organ, and is quite separate from the rest of the organs in the human body. So I think that's one reason maybe why it doesn't often metastasize, but it also has quite a specific metabolism, which is kind of immune cold, and is very quite strongly driven by hormones, in particular, testosterone and androgen. So I guess the kind of the environment might be quite controlled in the prostate compared to other other organs. So I don't know. I don't know if that means that just the drivers are not able to establish themselves. The cells with drivers that maybe drive cancers in other organs are just not able to establish themselves within the prostate because it's such a controlled environment. I don't know, to be honest. But we do acquire, a lot of mutations, do occur within the prostate, and a lot of copy number events as well. But most of them seem to be don't actually drive aggressive cancers. So it does appear that there's maybe quite strong selection against acquisition of strong drivers in the prostate.
Thank you.
Any other questions from the audience? Maybe a quick one, just to finish on this topic, maybe to both link to both Pierre and Claus' presentation. How early, maybe Claus, do you think this kind of normal cells containing driver mutations would have an impact on the immune response? So how early these normal cells would have some immune cells around?
So certainly, in the studies that have been done, there are very early changes in the local microenvironment. So speaking, from a pancreas-centric perspective, for example, when these cells acquire oncogenic mutations in KRAS, a lot of them start secreting GM-CSF, for example, and so you very quickly see changes in the local immune microenvironment. So I would absolutely suspect that at these early stages you would see changes, and one of the things we're kind of I think would be interesting to think about is for early detection purposes in particular, would you be able to distinguish tumors that are more poised to undergo transition into an adenocarcinoma based on the local microenvironment? Because that would be a certain a transcriptional state of that tumor cell that would then be reflected in what they are able to recruit, and whether you can use that as a distinguishing feature. I don't know. I think for me it's an interesting question.
If I can just add a little bit to it, it also depends not only on the tumor mutation, on the driver mutations, because a driver mutation at best will provoke one neoantigen, but it depends also how many passengers there are. Some passengers may have some mutations that can create neoantigens, although they don't really affect function and don't give an advantage. And therefore, a cell with one driver might elicit less response from the immune system than something with a lot of passenger mutations that actually are recognized. And I think there's an MSI subtype in pancreatic cancer. And I guess these tumors should really early on provoke an immune response, and probably have to learn how to go through that immune response very early on.
And it's a very good point. I mean, so pancreas cancer is, generally speaking, incredibly immune cold. I mean, immune checkpoint inhibitors have been tested in pancreas cancer patients and have failed, except for MSI patients that seems to have a bit of response. So I think your point is very well taken. And what's also interesting is that so people have also started looking at the type of antigens and predict, you know, based on the mutations, what are the types of antigens that might be expressed. And it actually looks like it's not just whether or not there is a particular antigen, it's also whether that antigen have a particular, you know, similarity in particular with pancreas cancer to bacterial or other sorts of, you know, antigens presented, and the similarity there seems to also be associated with the effectiveness of the immune infiltrate.
Okay, thank you very much. I think we are a little bit already over time. So what I propose is to maybe stop here. Thanks again to all the speakers today and the discussion and the stimulating questions we had. I don't know if the organizer wants to say a word to close up the session after that, or if we are all good.
No, it's okay, Mathieu.
Okay, perfect. So then thank you very much, and maybe see you in the next session for those who will be coming. Bye.
Bye-bye.